Also thank the organizers. This month's fellowship has been extremely enlightening for us experimentalists. We're moving from heterogeneity in the microcirculation to heterogeneity in the larger vessels, including the coronary arteries. But also I'll be talking about outside of the coronary vessels where actually most of the work has been done. And relating it to flow and fluid dynamic properties in large vessels at the macro scale, that is regional heterogeneity of endothelial phenotype. Now, the coronary circulation, like the systemic circulation, is very complex geometrically. And as a consequence, the fluid dynamics going on and the flow profiles and characteristics at a number of sites which near branches, bifurcations, curvatures, induce flow separation, which creates regions, transient regions of flow reversal and very complex flow. And these regions are very prone to atherogenesis. The proximal coronary arteries, for example, and also near branch points, where, for example, where the circumflex comes off from the LAD. And in the systemic circulation, particularly the internal curvature of the aortic arch, where there's flow separation, and the aortorenal junctions, and in fact many junctions, are all prone to develop atherosclerosis. Now, these are susceptible regions in the normal arteries. They're manifest basically as different regions of flow without any pathology being present until the pathological conditions develop. And a really intriguing question is what confers susceptibility to these regions, and what role does the endothelium play in fact mediating the pre-pathological susceptibility. So that's really the content of what I want to talk about today. Now, other regions shown here in green are actually spared. The classic one is the unbranching common carotid arteries, where indeed it's a simple tube and can be treated more like a Purcell flow type of situation. And these are very useful for comparison. Now, a picture's worth 1,000 words, and David Steinman has done some very nice, this is a carotid bifurcation, here's the carotid sinus, and I think you can see just past peak systole that there is a clear flow reversal going on only in this region. And indeed, that is a region which develops atherogenesis. And this is not any pathology at present, but clearly you can see the beginning of flow separation at the top of systole. In the coronary circulation, this would be reversed, would be in diastole for maximum peak flow. And what you see is that everywhere there's very nice pulsatile laminar flow, which is unidirectional. But here we have a flow reversal which breaks down quite considerably. It's still probably laminar flow rather than turbulence, and then reestablishes. And this occurs every second. So clearly, the question is, are the endothelial cells in these regions different? And how different are they? And is that related to susceptibility to atherogenesis? So if we consider the reasons for the transduction, if you like, and transmission of forces in these locations to the lining endothelial cells, we're really dealing with flow characteristics and a spatiotemporal variability of shear and shear direction and shear stress, of course, which includes changes of transport of solutes coming both out of the vessel wall as well as those in the blood. And we're also talking about the direct deformation properties of shear stress in particular on the endothelium in these locations, which becomes very complex and very different in these locations versus adjacent protected regions. I won't get into the detail of that, although I spent most of my life trying to dissect that. What I want to do today is really to sprint my 12 minutes to try and get a few points across, which might be at some point incorporated into the cardiac physiome modeling. Just to remind you, the endothelium principal functions, it exists really as a blood-compatible container. The blood would clot if you did not have an endothelium. And indeed, anticoagulation, both passive and active, are responsible for that. It's also a very important regulator of acute contraction and relaxation of arteries, as well as the chronic remodeling of arteries, which goes on when biomechanical environment changes over a long period. It regulates the permeability between the blood, and this is very important, and the vessel wall. This is very important for regulation of transmural transport. And it, when called upon, facilitates the inflammatory response as really the leading edge between the blood cells and which are involved in inflammation and need to be attracted to create an inflammatory response, which is one of the early hallmarks of atherogenesis. So basically, the arterial endothelium in a high-pressure, high-flow environment coordinates adaptive physiological and pathological responses by its malleability, its plasticity, if you like. What we are interested in these regions of flow separation is a chronic, steady-state phenotype change, which is a reflection of a chronic plasticity, if you like, of the endothelial cell. So how do we approach that? Well, we're basically saying that within the constraints of its fundamental properties, an endothelial cell always has these fundamental protective properties. There is also a malleability which is modified in part by its contact with the blood and the local biomechanics that go on at that interface. And so at different scales in vivo, we can address endothelial cell phenotypes and the heterogeneity, and in this case, regional heterogeneity. Now, the cells themselves actually reflect these separations of flow. Most of your arteries are covered with beautifully aligned endothelial cells like this. The junctions are shown here with silver staining. And this really reflects the directionality of a normal pulsatile laminar flow, always in the same direction without any reversibility. But in regions near branches, as shown here in a baboon descending thoracic aorta near an intercostal branch, you can see over a distance of 1 or 2 cells, the alignment shifts to a polygonal morphology over a very, very short distance. And this is a region of flow separation, and the cell morphology reflects that. And that's a very nice guide for us, because when we experimentally go in and take these cells and ask the question, how different are these cells phenotypically from those? These are susceptible to disease, these are not. Then we can actually confirm that indeed we are choosing and isolating cells from the appropriate region. So our strategy is to use a high throughput approach combined with reductionist science, which I'll get to in a moment, to use the genomics to inform us as to what kind of pathway analysis we need to do and which proteins we need to go after and post-translational function that we need to go after in these locations, but guided by the high throughput genomics, by isolating cells from these regions, isolating very small amounts of RNA, amplifying the nucleic acids, doing microarrays. The amplification must be linear and it has to be very rigorous, and then mining that data for guides to do reductionist science. Now, the first elements that we really analyzed thoroughly were the internal curvature of the aortic arch versus a descending portion of the thoracic aorta, where flow was unidirectional. So it was a comparison of a disturbed flow region with an undisturbed flow region. And postdoc Tony Passerini has a number of papers, a very nice paper in PNAS several years ago now. But what we discovered from this was that the endothelium in the disturbed flow aortic arch was clearly different in terms of a number of aspects of its phenotype compared with the descending thoracic aorta. And some of these are listed here. And what we found in most cases was that each of the categories of change actually included a balance. And this refers back to what Sasha was just talking about in the context of balances, checks and balances really being important for angiogenesis. There are checks and balances for many of these processes which have been modified in these specific regions of flow disturbance. We found both pro and anti-inflammatory phenotypes, groups of genes and proteins which in fact were working both in a pro-inflammatory sense, were primed for inflammation, yet there were anti-inflammatory pathways also upregulated to counterbalance that. And indeed, there's no pathology in those regions. So these cells are in a different state, phenotypic state, which we, in a very simplistic way, regard as a susceptible state. Elsewhere, no lesions ever develop when we do a comparison. We really regard this as a protected profile. What we find in the disturbed flow regions is a susceptible profile, and what we find clearly and are experimenting with now is to drive this susceptibility towards pathology and see how the phenotype contributes to that. But really, this is a different definition of susceptibility. It's a balance. It's not a threshold so much. We don't know if there is a threshold and when it kicks in. And I think it's an important general consideration. Now, we've extended this recently to go into 13 sites, including the coronary vessels and the arch and carotids and so on, and the aortorenal branches, to try to find some general principles of what we're finding different in the disturbed flow regions versus their nearby undisturbed flow regions. And we go directly into a large adult swine model and isolate the endothelial cells from all these different regions. and then we go through the microarray procedures and molecular biology, and bringing in very sophisticated bioinformatics for differential gene expression, such as page permutation algorithms using a false discovery rate methodology, gene ontology analysis, which tells us classes of gene sets which are different in different regions, and analyzing those. And then going on to pathway analysis, which I'll get to in a moment. And out of this, we really find a number of what we think are important signatures, which may be related to each other. And they're the three that I want to finish off with to present to you today. This is an ongoing work. Some of it's published, some of it is not. And they are three signatures associated with heterogeneous phenotype and associated with all of the regional flow separations when we analyze the whole database of 13 reference regions of disturbed flow and undisturbed flow in the adult pig. And they are endoplasmic reticulum stress, which initiates an unfolded protein response, the generation of reactive oxygen species, particularly superoxide, and the differential expression of key microRNAs, small inhibiting RNAs, which are very, very important at the post-transcriptional level and pre-translational level in terms of regulating the RNA levels for genes, gene expression, and also inhibiting when they're expressed highly, inhibiting translation, and therefore proteins are not made. And I just want to very briefly go through each of these and then end. So what we found in the ingenuity pathway analyses and other pathway analyses as a very prominent and dominant difference in the disturbed flow regions in all of the 13 sites was an association. About 72% of our top ranking ontology analyses came through with protein folding and endoplasmic reticulum differential expression. These are all upregulated, and when you put them into a pathway, like with ingenuity, as Sasha showed, similarly using a different approach, you see all the interrelations between these genes and proteins. For that kind of interaction, it's highly unlikely that it's random, and indeed it turned out not to be the case. So we homed in and said, okay, we're guided by the genomics. Let's now go in at the protein level. Let's go in at the QRT, PCR single gene level and actually try to understand what is going on in terms of the endoplasmic reticulum protein biosynthesis. And this next slide summarizes essentially a rather complicated set of experiments which are currently in press. What happens when proteins are synthesized and they're shipped into the endoplasmic reticulum? They're shipped often in as unfolded polypeptides, and it's crucial that they're folded properly and before they go on to post-translational modification and export or use in the cell. When the cell is stressed, and this is true of all, it's ubiquitous to all eukaryotic cells, there is an adaptive response called the unfolded protein response, which steps in when the cell is stressed and is overproducing unfolded proteins to try to rebalance the situation for cell survival. If it's unable to rebalance it by the unfolded protein response, then the cell will go on to programmed cell death. Now, the key players in the unfolded protein response are an inhibitor of unfolded protein response called BIT, also called heat shock protein 8-5, HSPA5, which, when the cell is stressed, it dissociates from 3 sets of chaperones in the ER membrane. And when it dissociates because it itself goes on to try and bind unfolded protein and fold them properly. Then these elements are phosphorylated or spliced and activate these triple UPR responses. What Mete Civelik, who's just graduated his PhD, just last week went on to a postdoc at UCLA, was elegantly able to demonstrate is that when you look at the mRNA level of the players in this pathway, or you look at the protein expression or activation of the phosphorylated form of these pathways, 2 of the pathways, the IRE1 alpha, ATF6 pathway, splicing of XBP1 mRNA, these are activated. The PERK pathway is not activated. So the bottom line is that the activation, the key thing is the activation of the canonical regulating protein BIP or HSPA5 is clearly activated in regions of flow disturbance. So what we have is 2/3, if you like, or possibly more than that, but a partial activation of the ER stress UPR pathway in endothelial cells in these regions. In other words, the biomechanics or of the environment in these locations are acting as a classical stress response, just as super radicals would, for example, or other cytokine stress and other kinds of stress. So what we're seeing is an adaptive response to the stress environment. Now that just is summarized here, I won't go into that. The second point is the fact that the reactive oxygen species, which is a functional outcome and you can test directly by dihydroethidium staining, here shown for 3 examples in a disturbed flow region versus an undisturbed flow region in the systemic circulation, is clearly elevated in the disturbed flow region. The red nuclear staining is the intercalation of oxidized dihydroethidium, which becomes ethidium and intercalates into DNA. So what you have here is a functional readout clearly demonstrating that reactive oxygen species, superoxide anion in particular, are elevated in that situation. The problem which we're struggling with right now, which relates to the coronary circulation, is it's not as clear-cut in the coronary circulation. So here you see a disturbed flow region, the proximal LAD versus a distal region. Yes, there is more in this case. In this case, however, there's not much difference. In a third example, maybe it's up. But if you look in the proximal right carotid, again, disturbed, undisturbed, you sometimes don't see much difference. So there's a lot more variability in the coronary circulation. However, the fact that these differences exist are clearly important functionally, because if you have a lot of superoxide anion around, it's not very good for the endothelial cells. Whether the superoxide anion actually creates the stress, which we are showing with the ER stress UPR system, we don't know, because in fact the superoxide production can be a consequence of UPR. So it's chicken and egg, it's a circular system. We don't know which way around it is right now. The final example I want to show you is the small microRNAs, which we find that they're some of them are highly suppressed in the disturbed flow regions. And when we go in there, we find that indeed, and analyze in this case, MIR10A, it's clearly much lower expressed and suppressed in the disturbed flow region than the undisturbed flow region. And if we do a spatial analysis in the aortorenal junction, we see a gradation of undisturbed flow to disturbed flow, in which you see a very nice gradation of expression, or suppression rather, of the MIR in question. One of its principal targets is Hox genes, which are homeobox genes, and you see a reciprocal relationship, as you would express, since they suppress Hox genes. When MIR10A is up, the Hox gene is down, and so on. And again, you see this very nice relationship. These are very important regulators. There are about 3 families which we're investigating. And finally, leading into this and possibly linking this together is that the NF-kappa B pathways, when we knock in and knock down the MIRs in question in cells in culture under very controlled conditions, we are seeing the reciprocal, basically green and red differences, that you would predict in a number of key molecules in the pathways for NF-kappa B inflammatory pathways, which may in fact be linking all of this together. So in summary, it's very simple in general principle that to use genomics of the endothelium in a spatially defined way, it's really discovery science, which often gets a bad press, but it can be used very, very nicely to identify where to look to do your reductionist experiments, and then go ahead and do conventional nucleic acid and protein biochemistry, the complementary approaches which each informs the other. And finally, I acknowledge not only members of my lab, but the Center for Bioinformatics, very close colleagues who work with us on this, and for the large animal studies, the US FDA Center for Radiological Health near Washington. And these are a group of very talented individuals in my lab. I single out in particularly Mette Civelik, who's for the UPR work and ER stress work, Yun Fang and Kangzu Shi for the miRNA work and ongoing work, and Elizabeth Manducci, who's a fabulous bioinformaticist and a very close colleague. And this is done at my institute at Penn. Thank you very much.
We are over time, but there's just one quick question while we change speakers. Quick question. Okay, I'm sure there'll be